will be Hannah. She's an assistant professor from University of Wisconsin-Madison, and she has taken a deep dive into both experimental data and machine learning methods, and using them to model protein dynamics and study how they influence protein function, how did they evolve over time, and she has also conducted quite some interesting interpretability study. Let's welcome Hannah.
Thank you so much, Gideon. Thank you so much to the organizers. Very happy to be here. This is a really exciting day. Let's see, so green button goes forward. Okay, great. So I'm obsessed with protein dynamics, and I kind of got into them from, like a geeky way, but I want to start by trying to communicate why protein dynamics matters for so many problems that we care about and that are really current, outstanding problems for us. And so a lot of the problems, and so when I say protein dynamics, I mean time in biology. This is fundamentally, if there's one word to take away, it's that time really matters. And so there's this paradox related to this really high-profile drug named Gleevec. So Gleevec was a massive success story for pharma. It completely reversed patient outcomes for a form of leukemia. And so, let's have a laser pointer here. Great. So here's Gleevec bound to two structures, and these two structures are from the PDB. And if you were to look at these structures a long time, and if you were to look at what residues are binding Gleevec in these two structures, you'd realize that they're actually the same picture. And so biochemically, the residues that bind Gleevec in both of these are completely the same. But then if one measures the KDs, and the reason why Gleevec was so successful as a drug is that it has nanomolar affinity to this kinase named ABL, and there's related protein human kinases in the body, such as this one SARC, to which its affinity is 3,000 times weaker. And so this was a big paradox because Gleevec actually was kind of discovered by accident. It'd be amazing if we could actually design things with this sort of specificity. And so a lot of people, a lot of kinase experts worked on this for a long time, and this paradox ultimately came down to kinetics. So it came down to time. And so it turns out that there's two steps to how Gleevec binds, and that these two kinases have different timing in how those steps work. And also, just to point out how our current, state-of-the-art KD predictors do on this, so Boltz-2 doesn't recognize that there's this 3,000-fold difference. So these are pretty much within error of each other in these predictions. So taking a step more broadly, this is kind of the geeky reason why I think that dynamics is so amazing. So to me, dynamics exists because evolution had to design computers. And I'm very happy to come after Bowen's talk because he highlighted how this can work at a big level for big operating things in biology. But the really kind of beautiful thing to me is that as people have gone and realized that we have these molecules and they need to switch between these different states, these off and these on states, what people have realized is that actually if we take one of those proteins, even just one, and then we look at it in the absence of the other effectors that it might be binding to, that in that protein itself existing in a test tube in the absence of everything else, it actually already contains information about those states. And to me, that's a crazy and kind of beautiful thing about biology. And that's something that now, has taken a lot of experimental work to figure out. And so another kind of distinguishing takeaway of this idea, if you've ever taken a biophysics class, you might have heard of this as conformational selection. But this is just the idea that the molecule has to switch to the on state itself before binding this effector. And that the barrier of going between the off state and the on state has to involve a real information-rich switch in what's going on. And so that process is often slow if we're thinking about comparing to thermal fluctuations. So in these basins, there's all these thermal fluctuations going on, but there's this really information-rich change that has to happen. And so some examples from my own study. So this is just showing, this is molecular dynamics of this metamorphic protein, Chi-B. So, it's wiggling and jiggling around in these barriers, but then a really interesting thing for Chi-B is that it itself, even in the absence of its binding partner, where that as part of its function, its on state is binding to that binding partner, but this itself, over a course of hours, unfolds itself and refolds to this. So this is what's known as a metamorphic protein. They're a little niche, plenty of proteins don't have a structure change like that, but the same principle holds for a lot of other things we care about. So for instance, this is enzyme, adenylate kinase, where the rate-limiting step of that chemical step is this interconversion between this open state and this closed state. And so here this process takes place, this concerted process of changing at about a millisecond time scale. And so this is a pretty big scale conformational change, but these concerted changes don't necessarily have to be big. So another example is this NTRC receiver domain, which is involved in signaling. This is just this little change. This helix just kind of gets rearranged, but there's these side chain processes, and so this interconversion also takes place on a millisecond process, and we can think of it as this switch between this off state and this on state. So I thought this was all super just amazing and cool and beautiful, but a question that I had was, how do we know any of this? You know, I can show you these simulations. So at the start of my PhD, I was in a molecular dynamics lab, but at the end of the day, that's just in a computer. And so something I kind of felt embarrassed I didn't know, what are the experiments by which people actually figure this stuff out? And in the name of machine learning as well, and as this is a new frontier, I think it's really essential that we figure out what we want to compare to. And I'm going to talk about some things that I think are kind of increasingly being used as a ground truth for dynamics, but which I want to just lay out some reasons why they're not. The first one, simulate, as I just said, incredibly useful tools, but the MD experts themselves need something to compare to when they're coming up with their methods. Heterogeneity in the PDB is also a really interesting signal to start from, an interesting hypothesis driver, but it itself, the number of counts or anything is not going to be the same as a free energy landscape. And then, so there's this funny type of structure in the PDB, these NMR structures, where there's this kind of spaghetti. And this is, I've been seeing an increasing number of bioarchives that use this as an indicator, you know, is this dynamics? Is this? And I'm sorry to say that it's actually not. And so folks besides myself have tweeted on this. So Preston Lindorf-Larsen likes to talk about this. So the NMR community has not done a good job of communicating this. This is modeling uncertainty. And so I could be a very bad actor going forward if folks keep using these. I could just deposit a bunch of crappy NMR structures in the PDB and probably have like a net bad you know, influence on the state of dynamics going forward. So in Dorothy Kern's lab, in my postdoc advisor's lab, I had this question, where does one find like a ground truth for for dynamics? And so the Kern lab studies how dynamics leads to function in a variety of systems. And the Kern lab uses all three of these structural biology techniques because dynamics is present in all three of them. But for that question I had about time, where does one find time in biology? She encouraged me to spend some time with this guy at the bottom of the pool. And so, but wait a minute, I just told you NMR structures are not dynamics. So what the heck, what's going on? So this meme actually doesn't do a good job of representing NMR. So there's a bajillion things that count as NMR because there's a bajillion ways you can shoot energy at a sample. And so I fixed this meme. So here's the updated meme. So there's a bunch of things at the bottom of the pool. And so the part of NMR that I'm really excited about, I view as a future that is complementary to a lot of other techniques, is this relaxation dispersion NMR. And so this is what can give us quantitative measurements of kinetics in populations. And so as an example of this, so if we take that NTRC domain as an example, the way that people figured out that there was this allostery in this NTRC domain was that they could do one of these relaxation experiments. And a common thing is to plot this value of this observable, this REX, to plot this as colors on a structure. And so even if you don't necessarily know what that slow timescale process is, if you do this REX experiment, you see, oh, this side of the protein lights up. So if I've done my experiment right, that means I know that part of the protein is involved in a millisecond process of some sort. And so as I was, so I then also want to try to, before the rest of this, just tell you a little bit more about NMR, not drawing on quantum at all, because I'm still a little bit scared of quantum. But there's other analogies that I hope can just kind of communicate how this experiment is working. So in an NMR experiment, so I'm showing you here a protein backbone. And so what I need to do is I need to have atoms that are magnetically active. So happy to chat about it later, but there's interesting physics as to why this is true. But so the most abundant form of hydrogen in the universe is already magnetically active, so thank goodness. All then we have to do is put in a magnetically active nitrogen. And what I'm showing here are these atoms as handbells. And so this is an analogy that when it was first told to me, I just thought was so cool. And so because then it turns out the resonant frequency of each atom is dependent on the environment that it's in. And so you can kind of think of a protein in an NMR experiment as a set of handbells. And so then what we have is you see these 2D spectra, and really each of these axes is a frequency. Like if there's music geeks in the audience, we can really think of this as a series of pitches. So these things down here have a lower pitch, these have a higher pitch, and this is what allows NMR to be such a sensitive readout to all sorts of effects that we might want to look at. And then, so each of these peaks here corresponds to one of these sets of nitrogen and hydrogens in the backbone. And so this is fundamentally time information that we're measuring and then doing Fourier transform to get this sort of thing. And so then also, if there's dynamics present, even these simplest experiments contain information about it. So if, for instance, in my Chi-B protein, if I'm interconverting between one state and another state and my chemical environment is changing, here maybe a handbell isn't a good representation anymore. Maybe it's more like a violin string or something that I'm changing the pitch of. And so if I'm going from, B flat to G flat, depending on what rate I'm doing that at, my signal is going to look different. And so if the process is slow enough that I'm going between these two different frequencies, I'll see them as two different sets of peaks. If it's fast, this is maybe also where the music analogy breaks down because our ears don't really do this. But if it's too fast, then these average into one signal. And if it's in the middle, then I get this broadening that can happen. And so then these relaxation experiments are, and even the simplest one, we have information. The relaxation experiments allow us to more quantitatively monitor basically the dynamics that are going on. And I can think of it as kind of ringing this bell and then quantitatively monitoring the decay of it. That's where the relaxation term comes from. These are the only NMR equations I'm going to show you. But so this is a common experiment that's done of collecting these two different rates that reflect both when there's picosecond to nanosecond motion and when there's millisecond motion. And so a way that an experimentalist often plots this data, so this is the same data as in that NCRC protein, they'll often plot a ratio and then look for where there's spikes in this ratio. And where those spikes are is where there's this relaxation term due to where that millisecond motion is, that longer time scale, possibly concerted motion that we're interested in. And so I was learning more about this. I was realizing that these experiments have been being done for the last 30 years and being plotted often as colors on structures. And as someone from a comp bio background, I just thought, well, we got to make a benchmark of this. This seems really useful. Unfortunately, they've never been required to be deposited or anything. And so then the question was, how much of this data could I curate before getting tired of this endeavor? And so the answer, very concretely, was 133. So RelaxDV is a data set now of 133 different proteins selected to be in apo form so that one could really associate whatever dynamics is in there with what intrinsically is in the protein itself in its apo landscape. And happy to chat through technical details. That was kind of hard to do. But so this is what the raw data looks like. It's all these R2, R1 traces. And there's numerous annotations. And there's annotations as well when there wasn't data of where these different sources came from. So here, this is just, when I go to conferences to other people who have big data sets, I like getting to tab through this and show it's only 133, but it's still maybe orders of magnitude more in one place of this than we had before. But 133, I can't really train a new model from, of course. Like, that's not going to generalize. I was sitting in my postdoc advisor in Dorothy Kern's office, kind of feeling bad for myself that I only had 133 to do anything with. And she pointed out, well, okay, you gathered all that data and curated it and everything, but that's not the entirety of the residues. The residues for which you could get those traces to measure it, the relaxation, that's not everything that has exchange or that has that slow motion. So she pointed out there's some residues that are missing from the spectra entirely because they had motion and the peak happened to just be completely broadened beyond detection. And so then I this led me to go back and also then go back and figure out, okay, which of the residues that I don't have the relaxation data from, which of them might be missing due to some sort of experimental thing versus actually being completely missing from the original assignment information. So the assignment information is the one thing that was ever required to be deposited for NMR data sets. And that is what allows me to say in this data which of these peaks corresponds to which residue. And so what was kind of interesting in this data was that if I made this plot of sequence conservation, you can particularly see this in core residues. So I have these different categories of residues with picosecond, nanosecond motion, millisecond motion, and then also where amide assignments are missing from the assignment data, that both the millisecond motion residues and ones where there was amide assignments missing were both more conserved than my category of residues with no motion label and ones with picosecond, nanosecond motion. And this is really interesting. I really didn't know what to make of this. It's not a huge effect, but it's definitely there. And so I thought this was maybe interesting and kind of surprising. But then as a postdoc at Brandeis with many distinguished enzymologists, I was showing this on a poster at one point, and some people said, well, duh, that makes total sense. Because I study some allosteric process or some binding process where I know that the residues that are involved in the function of my protein are very conserved, and that's also where my millisecond process is happening. So of course the residues that are related to function are going to, that have those dynamics are going to be more conserved. So I just thought it was interesting to be able to see this with only 133 proteins. But then the more, perhaps a more interesting thing, I kind of got goosebumps to realize this at a certain point, was that, wait a minute, you know, these relaxation experiments, it took a lot of work to curate 133, but there's actually data for thousands of proteins for just the assignments. We have data on the missing assignments of thousands of proteins. And that's all from that data that was required to be deposited in the BMRB. So the BMRB is the little kid sister of the PDB, but for NMR. And so at one point, then I thought, what if I just try to curate out all of that to make a, quote, missing BMRB data set, and if a model could be trained on that, could it have predictive power beyond just what's missing, but to actual dynamics? And so the way that this was then done, so there's 12,000 proteins roughly in May 2023 that I could use for this. For this data set, I removed samples for which there's a variety of other reasons that assignments could be missing. This still left us with about 9,000 proteins, and so then I devised this labeling where we are considering where there are assignments missing that are basically between the first and last assigned residues. And even with that in consideration, even for these small proteins, there's a substantial number with amide assignments missing. And at this point, I was very excited to meet Gina, actually via both of us helping organize MLSB, because there was just a bajillion ML problems in front of me with this task, and was just very excited that she was interested in this problem too. And so, because now we had this kind of very, posing dynamics as a very crass binary classification problem. Can we predict which assignments are missing? And so we combined this missing BMRB data set as well as these more information-rich curated data sets, split these in just conventional, was now conventional for protein ML ways, and then we have a test set, and then also critically these more well-understood validation sets to test any models on. And so what we found, to make a long story short, is that we found that fine-tuned versions of ESM models, as well as things built on AlphaFold, had higher predictive power for this task of predicting missing assignments than a variety of other baselines that we considered, including things like normal mode analysis, which is a very old way that works for some things of looking for those concerted motions. So this is on the BMRB test set. If you saw that slide before and saw that there was an 80% sequence split, you might be screaming, because that's like not a very good split for a lot of protein questions. We tried going down to training data size that was much smaller, doing a sequence identity split of just 30%, and also removing anything with a structure homolog. So at the end of the day, this model and all the models I'll talk about going forward were trained at this most stringent training split. And so this ultimately meant it was only trained on about 3,000 proteins. And so this was quite interesting as well to see that it suggests to us that there was a lot of signal already in the ESM layers towards this task. Interestingly enough, also though, if you then go up to 8,000, the performance in this generation of models really doesn't change that much, suggesting there's a lot more capacity for a model to do better at this. And so then these predictions from this binary classifier, we converged on visualizing these as this putty representation, where so the model is predicting the likelihood of an assignment being missing. And so fat sausage is a p missing of 1, and a skinny sausage is 1 of 0. And so here's one of the ones from our test set. Again, no structure homologs, no sequence split to 30%, where here I'm showing in red where the peaks were actually designated as being missing. The authors noted this. And then in gray is where peaks are present. And so then what was really interesting was generalizing beyond just the data of missing assignments. We observed that this model had predictive power for measured exchange in the set of these relaxed DB proteins. And something that we thought we noticed was that it also seemed that like where authors had sort of identified a motif in the protein that was dynamic, for instance, here this dynamics in this active site of this mercuric transport protein or this FMN binding site of this flavodoxin, those seem to be examples that the model was doing, had predictive power for. And so a test set that I felt very, I really wanted to be sure that we tested on, there's these, this kind of the big guns of NMR is this CPMG experiment. I can only curate 10 of these experiments in total, which I think is a real shame because there's more of them out there. But there was an interesting reanalysis of the data that we could do. So all of these are systems that have been studied a lot and also been shown to have these concerted global processes that are underlying that transition. And a way that one looks very concretely is so this is what the raw data looks like, and if there's a dip here, that's indicative of REX. And so it's promising to see that this model had predictive power for those global concerted processes. Along the way, I realized that there's actually another indicator from the raw data that there can be exchange, and if I then reanalyzed, also taking that into account, the performance actually went up. So this allowed us to go back and reinterpret these raw data sets, and so this just underscores to me the essentiality that we have raw data for this going forward. But there's room to improve with this model, so we named this model Dino1. So, and this NTRC protein gives us a good example. So the model had predictive power for exchange that's known over here, but there's this tyrosine residue here that's also known to have slow exchange, but the model doesn't predict anything there. And something that's kind of a shame, so this is a spectrum of NTRC. This is the simple spectrum, the simple experiment, but you can already see in this simple experiment that it has exchange because the peak for tyrosine 101 is really small. And so an NMR experimentalist could gather this, assign it, know that's tyrosine 101, and be like, of course it's going to have exchange. And so, but the shame is that these spectra were never required to be deposited anywhere. And for a long time, this kind of just seemed like it was going to be a total no-go for someone wanting to do ML with NMR if we're only limited to the possibly imperfect assignment data sets that were required to be deposited. And so just kind of had this dream, but what if we could just scrape these off the internet? Because often those 2D spectra are put in papers as a kind of quality control measure. But I just had no idea where to even start with that until one day Gina and I were on a Zoom kind of just speculating how could one do this? And we realized that there's this little journal, this biomolecular NMR assignments journal, and figure 1 of all of them is one of these HSQCs. And so then we're like, wait a minute, like what if we just like got all the figure 1s out of this journal? Could we start there? And what would it even mean to do that? So Gina took this on. She tried starting by, so we downloaded all the ones from that one. She then went and got another list of ACS journals. She got cut off and got a cease and desist email from the Stanford libraries. So we tried to figure out what our other options might be. Really fortunate to get in contact with Ian Ross at Wisconsin, who's been integral to this project from the geology department, actually, this project called Extraction of Dark Data, where they've come up with a bunch of journal agreements to be able to have access to like huge PDF stacks. And so from that, so there are 16,000 current BMRB depositions. Gina, with Ian's help, narrowed this down to about 10,000 viable protein depositions. And then from this could get 4,000 HSQC images. And then after some filtering, now has a set of 3,000 of these HSQC images. And so I have some pictures that I hope to just impress upon you. This is an unprecedented collection of time data. And I just get goosebumps from this. But the other thing to point out is that there's never been any standardization for how these should be portrayed, and these were never intended to be used in this sense. And so we have all this information, so the question is now, what do we do? The best that one could hope for is to find some incredibly talented and resourceful individuals, so namely Gina and an undergrad who's been working with us, Nevin Pai from Wisconsin, who then just set about trying to come up with a pipeline to, from these PNGs, get back out the peak information. And so, unfortunately, AI was not helpful. So Gina tried uploading an HSQC. It seems like a very clear task, right? We thought maybe with AI this could just be solved immediately. But this is not even the same HSQC. So I'm actually curious where this came from. And so what they did, they had to do some just old school data engineering, and so ultimately figured out a way to align coordinates from each BMRB entry to the images to then extract and use the segment anything model to get out information about each of these peaks. So through a tremendous amount of work, they now have these sets of masks for these HSQCs. Again, so where each of these, the shape, the size, all of this contains information about time related to these data sets. And we just now have this unprecedented data set that again, I just am so incredibly excited about. And as an example just of what we are able to pull from this, so just quick cursory analysis. Here's an example of a protein where in the HSQC you can see that some of these had exchange broadening. And if we just take the peak, the area, just a very simple property of this data, and plot it on the structure, we see that the residues with peak broadening are all around a particular part. So this is already an example of a previously undescribed millisecond motion that we could just get out of what was in the literature already. And so with that, I'm at the end of my talk, and I just want to leave you with some questions, which is just, one, what can we dream now of doing with this data of these thousands of actual fingerprints of this time information in biology? The next question, can we imagine scaling this to collect new dynamics data? So a student from my lab, Jojo, is here with a poster on this, and I really encourage everyone to talk with him and see the awesome things that he's been coming up with to try to do that. And then my last question for you. Please talk to us if you'd be interested at all. And with that, I'll end there, and super fortunate to work with these incredible individuals, and happy to take questions.
Great talk, Hannah. So instead of asking you about dynamics, which is super exciting, I want to talk to you about collecting data. So I think that we can all argue that none of us would be sitting in this room without the PDB. And you've clearly shown that the fact that this data has not been collected in sort of a routine way has at least, slowed down our ability to really use it. And so my question for you is, as we sort of think about moving forward and with all of these new high throughput, both computational techniques and then sort of pairing it with high throughput experimental techniques, what are some important lessons that you've learned from going back and looking at this data that has not been routinely sort of collected and annotated that we can really learn for what we need to do moving forward to be able to collect and annotate this data?
I guess in my area, the biggest lesson has been that we should really just try to dump as much raw data as we can. That's something really easily said in NMR, where it's only ever time series data. So the raw data is a lot smaller than other modalities. And I think something that, I think we ought to be able to leverage AI for is to say, I think a barrier to just dumping raw data is that it's a huge time sink on the part of the experimentalist. To, like, for instance, the current deposition process for the BMRB, there's probably like 200 metadata fields that one, in principle, should be filling out, and it's just like too much of a burden to fill all those out. And so I'd love to see something where you just, like, the upload process is you just point your magnet console to a URL somewhere and just say, like, figure it out. I think this should all be very possible. And I think that where I see communities getting hung up right now is on, like, what, we agree that the metadata is important, or we agree that formatting is important. And I see people getting hung up on those things, where I think, what if we just, like, dump everything in Zenodo right now, and in a couple of years we'll let AI stuff just, like, figure it all out. I don't know how much that generalizes to every data type, though.
Thank you for the nice talk. I have a question about the missing peak in NMR. So, in X-ray crystallography and cryo-EM, there are also residues that's not resolved in the structure. Have you ever looked at if there's correlations between those two methods? And
Absolutely did. And it was super interesting, because this is also a question, because also, like, AlphaFold has a head that was trained for that. And there's minimal overlap as to what has missing assignments and where there's missing density. So they're really complementary sources of information.
Thank you.
Thank you for the fantastic talk, Hannah. How important is it to be able to map the HSQC spectra to the information, like the frequency of the NMR spectrometer, the pH, the buffer, the temperature that these spectra were recorded at? So
something that we realized while developing the Dyna-1 model, there was I didn't talk about this, but there was a set of proteins where Dyna-1 actually had, like, worse than 0.5 AU rock. And that was something we investigated, what the heck is going on there? And we realized that a property of many of those was that they were RNA or DNA binding proteins that had been characterized in phosphate buffer. And so I, as the curator, didn't realize that that is something that could actually cause exchange of a phosphate weakly binding, unbinding. And so when we then removed those proteins, the performance of the model went up. So those sorts of, because these are, room temperature, super sensitive readouts, I think it does matter to have all those things.
Thank you.
One question is, like, do you need, like, the solvent viscosity recorded? That's something people argue about. I don't know if I have an opinion on that.
Hi, okay. Is this, I turned it off. Yeah, so I know, like, a lot of allostery occurs for, like, coupling dynamics between different regions, and you can have, like, binding or, like, domain insertions cause that. Have you, like, looked at all about, like, if your model can look at, like, coupling dynamics, I guess?
So several of the examples, like in this RelaxedDB and other places, are examples of allostery. So allostery is a catch-all for a lot of types of things. So depending on the system, I don't want to say yes or no to anything in particular.
Great. Let's thank Hannah again for the talk.
Thank you so much.